We left off in the last video with the decision acetyl-CoA had to make in the journey through our metabolic breakdown of carbs, fats, and protein. In this video, we will continue our descent through the metabolic pathways, through Krebs cycle, and electron transport chain. But first, let's do a quick summary of the last video. Glucose enters glycolysis where it is broken down into pyruvate molecules. Pyruvate is then converted into acetyl-CoA. With the triglycerides, the glycerol backbone will be converted into pyruvate. The fatty acid chains are broken down 2 carbons at a time to make acetyl-CoA. Amino acids enter the pathway in various spots but are largely converted into pyruvate or acetyl-CoA after going through deamination. Acetyl-CoA is the center of all 3 macronutrients. All of the carbohydrates, fats, and protein make acetyl-CoA. And the decision of acetyl-CoA is to either enter the Krebs cycle to make more ATP or store energy as fat. Assuming our cells need more energy, let's follow the path into Krebs cycle. This is often referred to as the tricarboxylic acid or TCA cycle, but you will hear it called Krebs cycle in most of your science classes, after the man who discovered it. The first thing to consider in the Krebs cycle is that it is a circle. Yes, we are starting basic here. So, going back to elementary math, where does a circle begin and end? That's right, it doesn't. A circle has no beginning or end, and the Krebs cycle doesn't really either. So, as we look at acetyl-CoA entering this cycle, it isn't really the beginning per se, but it acts with the compound preceding it, oxaloacetate. The only way for acetyl-CoA to enter the cycle is with oxaloacetate. The Krebs cycle goes through a series of steps, transforming one compound to another. Along the way, high-energy electrons are released. Just as we saw with the electrons produced in glycolysis and fatty acid oxidation, coenzymes will carry the electrons to the electron transport chain. In addition, carbon dioxide is released. CO2 also released when making acetyl-CoA from pyruvate. This is a fairly important concept because we know a certain amount of CO2 is produced with metabolism of each gram of our energy nutrients. This CO2 must then be removed from the body through our respiratory system, which is why metabolism is referred to as cellular respiration. Since CO2 produced is a direct effect of nutrient breakdown, by measuring CO2 exhaled, we can get an accurate view of the calorie needs of a person. This is actually one of the most reliable ways to measure calorie needs. We call it indirect calorimetry. As we go through the Krebs cycle, I mentioned that we release high-energy electrons that will be carried to the electron transport chain to make ATP. Krebs cycle is a critical step in creating ATP, so it is vital for Krebs to continue cycling. Remember that in order for acetyl-CoA to enter the cycle, the compound before it must be present. If oxaloacetate is not available from a previous step in the cycle, it can be made from another compound. Take a look at what else makes oxaloacetate. Pyruvate. You remember pyruvate from glycolysis. It is the end product of glucose breakdown. So the important concept here is that glucose facilitates the Krebs cycle, or more simply stated, carbs make the Krebs go round. Even if fat is being used as an energy source instead of glucose, glucose will be necessary for the complete breakdown of fat. This is another concept we discussed earlier in the semester. To break down fat, we still need carbohydrates. Now let's move into the final step of the metabolic pathways, the electron transport chain. This is where most of the ATP is produced. The hydrogens carried by coenzymes from the metabolic pathways enter the electron transport chain. Through a series of steps, the electrons are passed across the protein complexes in the mitochondrial membrane. Eventually, the electrons join an oxygen molecule to form water. The hydrogen protons are pumped into the outer compartment and eventually shuttled across a gradient through ATP synthase. The kinetic energy of the protons is captured in the phosphorus being added to ADP, thus creating ATP with stored energy. Now the ATP can be used to fuel processes going on in various cells throughout the body. I know this was a very long and complex subject, We need 3 videos to cover it all. But it is the basis of all the functions in our bodies. As we continue throughout the semester, we will refer back to the metabolic pathways to explain why we use certain nutritional changes to treat what is going on in the body. So when we talk about how we can prevent negative nitrogen balance during metabolic stressors, or why carbohydrate is reduced in respiratory failure, or how protein restriction can preserve kidney function in renal failure, we come back to the metabolic pathways to explain it.